In situ hybridization is used to detect the physical location of a gene on an intact chromosome. In situ is Latin for in place, and in this procedure the chromosomes are adhered in place onto a slide. The most common method of in situ hybridization uses fluorescently labeled DNA probes. This technique is known as fluorescent in situ hybridization, or FISH. To perform FISH, first cells that have been arrested in metaphase are treated to make them swell, and then they are fixed onto the surface of a slide. This also fixes their chromosomes onto the slide. Next, the slide is treated so that the chromosomal DNA is denatured into single strands. Then special DNA probes are flooded onto the slide. The DNA probes are small pieces of single-stranded DNA with a sequence from the gene of interest. Probes are able to hybridize only with their complementary sequence, which will be at the site of the gene of interest on a particular chromosome. The DNA probes are allowed to hybridize with the denatured chromosomal DNA, and any excess probes are washed away. Either the DNA probes themselves are fluorescent or they are chemically modified to allow fluorescent labels to attach to them, so that when fluorescently labeled molecules are added to the slide, some are able to bind to the probe DNA that is bound to the site of the gene on the chromosome. Excess fluorescently labeled molecules are then washed away. The slide is then viewed using a fluorescence microscope. The fluorescently labeled molecules reveal the physical location of the gene of interest.